This video offers a very brief overview of the taxonomy of viruses and how you can look at the name related to a virus and see what level of organization we're talking about. And then it's going to introduce the viral replication process. Please note that viral replication is the same thing as viral multiplication. And some also use the term viral life cycles. But all of them basically mean, the process of a single viral particle infecting a host cell and then using that host cell to make a lot more viral particles that then leave the cell to go on and infect new hosts. Okay, so briefly, the taxonomy of viruses, this is all about naming, okay? So if we're looking more, big picture, the order of viruses, a particular set of viruses ends in ALES, okay? Zooming in a little bit, family names end in Viridae, and zooming in farther, the genus name ends in virus, okay? So and then the species is going to be those groups of viruses that shares the same genetic information and has the same host, okay? So if we're looking down here at human herpes virus 2, all the human herpes virus 2s are going to have the same genetic information and infect the same type of host. You can probably guess HHV2 infects humans. Human herpes virus 2 belongs to the genus simplex virus. So again, notice here's the ending of virus, which is how you know that this is the genus name. Okay, and HHV2 belongs to the family Viridae. Okay, so you just need to remember that if you see Viridae, that's the family. If you see ALES, that's the order. If you see it ending in virus, that's going to be the genus. Okay, and that's as complex as you got to know as of right now. So then the next thing that I want to touch on is this viral multiplication or viral replication overview for viruses. And we're going to talk about the multiplication or replication process for bacteriophages, because remember, they only infect bacteria. So things are a bit different when the host is a bacteria compared to animal viruses, for instance, some of which infect humans. So kind of overall, though, big picture, the nucleic acids of viruses typically only code for a few genes. And remember, a gene is a segment of nucleic acid that codes for a protein. Okay, so if a piece of nucleic acid only codes for a few genes, normally that means it's coding for only a few proteins. And so it's a relatively simple virus. Now, there are viruses out there that do code for quite a few proteins and therefore have a bit more complex nucleic acid, but for the most part, they're pretty simple, especially compared to living things. So in order for the virus to replicate, it must, get inside of a host cell, and then it takes over the host's machinery, the ribosomes, the ATP, the nucleotides, amino acids, anything really that it needs to make more of its genetic material and make more capsid. It's going to take anything in the cell that it needs. In some cases, it actually destroys the host DNA so that it doesn't have to have any competition, which is pretty interesting. If you look at the picture on the right, we say that viruses have a one-step growth curve. Okay, so they're going to get in, they're going to take over the cell machinery, they're going to make new viruses, and they're going to get out. Okay, and so once we walk through the details of these processes, this graph will make a little bit more sense. But what I want you to notice, though, is on the graph, look at these axes, time in days, and then number of virions. Remember, y'all, virions means infectious viruses. They are complete, intact viruses that can go on and infect a new host. And so what I want you to notice, let me try and change the color of this pen real quick. Let's use blue. So notice at the very beginning, okay, there are some virions. Okay, we don't have a quantity along the y-axis, but that's okay. But notice the first thing that happens is the number of virions is decreasing, right? That's because once a virion gets inside of a host cell, it actually falls apart. It falls apart into individual capsids in its nucleic acid genome. And this allows it to then take over the host, because in order to copy the genetic material, the cell machinery must be able to access the genetic material, right? And originally the capsid is protecting the genetic material. So we need the capsid to kind of fall apart, so to speak, so that the genetic material can be accessed, so it can be copied, and so it can be used to make more capsid proteins, okay? So essentially the virions are gonna attach to their host cell, they're gonna gain entry, and then they fall apart. So when they fall apart, they're not intact and they're not infectious. Okay, so that's why this number decreases. During this eclipse period, this is when the viral genetic material and capsids are being mass produced. Okay, but again, they tend to remain as individual components hanging out inside of the host cell for a while. At a certain time point, all of the individual components self-assemble. Okay, it's called spontaneous assembly. And this is when they start to form more virions. Okay, so the number of virions increases once they all start to self-assemble, and then they push their way out and go and infect new cells. Okay, so again, here is where the mature intact new virions are being made, and then they're going to start exiting the cell. So again, those numbers are decreasing, okay? And we're gonna talk a lot more about this in future videos. The next video is going to be about the two replication cycles of bacteriophages. That's the lytic cycle and the lysogenic cycle. And then we'll move into animal viruses, which are a bit more complex. As always, let me know if you have any questions. I really need to think of a new way to end these videos. All right, y'all, have a good one.